Welcome to the Broad Institute. My name is Megan Rokup, and I'm the director of the Educational Outreach Program. And I'd like to welcome you to the third lecture in our series of 4 lectures of the 2012 Midsummer Nights'Science series. So today's our third lecture. There's one more a week from today, same time, same place, so I hope you will join us. It gives me great pleasure to introduce our speaker for tonight's lecture, Dr. Peter Reddien. Peter is an associate professor in the MIT Department of Biology, and he's a faculty member at the Whitehead Institute, which is right next door to us. And he's also an associate member here at the Broad Institute. Peter earned his undergrad degree in molecular biology at UT Austin, and he earned his PhD in the MIT Biology Department right across the street from us in that direction. He did his postdoctoral research at the University of Utah. Tonight, Peter will be speaking to us about the remarkable ability of flatworms to regenerate missing body parts. So please join me in welcoming Peter Reddien.
Well, thank you, Megan, for that introduction, and it's my pleasure to be here with you tonight. Thank all of you for coming and for your interest in this subject. I'm happy to tell you tonight about the work of my lab on regeneration. And regeneration is widespread in the animal kingdom and one of the most fascinating problems in biology. Observations and experiments on regeneration date back approximately 3 centuries to experiments by Remuer and crayfish and Abraham Tremblay and Hydra. In the intervening centuries, examples of animals capable of wholesale replacement of missing body parts have been found in almost every major animal phylum. Familiar examples include earthworms that can regenerate following being pulled in half, starfish that can regenerate new arms, salamanders that can regenerate new limbs, and planarians that can regenerate essentially any missing part of their body. So if the goal is to study regeneration, to understand how the process works, what we would like to do is understand the cellular basis for regeneration and the molecular underpinnings of the process. And we're interested in regeneration from a basic science perspective. It is a fascinating process, widespread in the animal kingdom, and we want to know how it works. But it is also of interest from a regenerative medicine perspective. There's a lot of interest in regenerative medicine using cells like stem cells, which I'll talk about some today, for the replacement of missing cells or damaged cells. And this process, all the goals of regenerative medicine work. Evolution has solved the problem in these animals, these regenerative animals. So I think it's our scientific task to try to understand the processes that allow regeneration to work in these animals that regenerate naturally. And then we can ask the question, what is different between these highly regenerative animals and other animals that are not quite as good at regeneration, such as ourselves? Now, to understand the cellular and molecular basis for regeneration, it's important to have a good experimental strategy. What would be desired are organisms that we could study in the lab that would allow us to ask questions about the cellular and molecular basis of regeneration. Desired attributes of such experimental systems would be the ability to regenerate robustly, ideally the ability to regenerate rapidly, the ability to culture the animals in the lab in large numbers, the ability to study the cells that are involved in regeneration with suitable methods, and finally the ability to study genes involved in regeneration. And today I'm going to tell you about our work on one organism that has these attributes. And that organism is a planarian. Planarians are flatworms and one of the classic model systems for studies of regeneration. So this is a single planarian with his head on the left. You can see 2 eyes in the head of planarians. These sort of cartoon-like crossed eyes are diagnostic of planarians. You can also see in the middle of the animal a pharynx and then the tail at the other end. Now, if you remove the head of planarians with an amputation, the head grows back. You remove the tail grows back. You take a tiny fragment from the side of the animal, and an entire organism will be regenerated. And this process of regeneration involves, in part, a process called blastema formation. Shown here is an animal in which the head has been removed, and a new head is regrown in about a week. And regeneration involves the outgrowth of new tissues at the wound site. Question?
Yeah, so what if we cut through the middle?
You could make a sagittal cut through the middle and produce two halves that would both regenerate their sides to generate two animals. Yeah. Your creativity is basically your limit with how you amputate these animals and look what they do. OK, so we have this unpigmented outgrowth at the wound. And you can see within it new eyes forming, and a brain is regenerating as well in this structure. Now, despite their seemingly simple anatomy, flat worm, there's a complex internal anatomy. Question?
Just out of curiosity, do you know the smallest piece that you can cut off, maybe percentage-wise, something like that, and have it still regrow?
The question was, what's the smallest piece that can regrow an entire animal? And the answer is a piece that has about 10,000 cells, which is roughly a 300th the size of a large mature animal. So the animals have complex internal anatomy that all has to be regenerated following injuries. For example, in their head, they have a brain, a variety of sensory cells like eyes and other types of sensory neurons. They're surrounded by an epidermis, as seen in this schematic cross-section of a planarian. They have a musculature for twisting and turning, but they move by the beating of cilia on the ventral surface, which are seen here. They beat and allow the animal to glide across the surface.
Cilia are tiny hairs?
Cilia are tiny projections off cells that can beat. They look hair-like, but they're projections off cells that can beat and cause forward motion of cells. So in the middle, you see in these two animals a centrally located pharynx, which is their feeding organ. And this connects to a multi-branched intestinal system, seen here, that delivers nutrients throughout the body. And there are dozens of other cell types that have been described in these animals. And because a tiny piece can regenerate an entire organism, there must be some mechanisms in the adult for the production of all of these cells, organs, and tissue patterns. How does that happen? To address this question, we use as a model Planarian species, Schmittea mediterranean. And these animals come in two types. On the left are sexual animals. And these animals grow to be about 2 to 3 centimeters in size. They're freshwater. And they are cross-fertilizing hermaphrodites. So they have both sperm and eggs, but they mate with one another to produce progeny. On the right are asexual animals. And these animals are in size of a range between 2 millimeters and 8 millimeters. And these animals reproduce by fission. So what will happen is the tail region of an animal will grab down onto a surface, and then the head starts crawling away from the tail, stretching out, until the animal breaks into two parts. And then both halves regenerate. Now, these animals are easy to culture in the lab. We probably have tens, if not hundreds of thousands of them in my lab. As you see and saw in the previous slide, we can keep them in Tupperware containers. We can also keep them in Petri dishes. For example, here, there are sets of animals in different Petri dishes. And in each set, the animals are undergoing a different experiment. So the ease of culture in the lab and the speed of regeneration enable a large number of experiments to be performed to try to get answers into how the animals regenerate. Now, the ability of planarians to regenerate has been known for nearly 2 centuries, and some of the first systematic surgical manipulations of planarians and observations were performed in the late 1800s, early 1900s by T.H. Morgan and colleague Harriet Randolph. T.H. Morgan is famous for being the founder of Drosophila genetics. Less known is his prior work on regeneration in planarians. And in the next couple of slides, I'll show you just a couple of surgical experiments that reveal some of the dramatic regenerative power of these animals and some of the dramatic feats that these animals can accomplish. OK, so in this experiment, you make an incision in between the eyes. And what happens is the animal regenerates 2 heads, such as this one. More complex manipulations can be done. You can cut the animal in any kind of sequence of events or transplant tissues. Okay, for example, in this experiment, there are 2 amputation sites One, the head was removed, and second, an incision was made from the tip of the tail up to near this amputation plane. And this is known to produce two-headed, two-tailed animals like this one. Now, despite decades of surgical manipulations and observations of Planarian regeneration, satisfying explanations for how it works mechanistically have been lacking. Morgan himself was once quoted as stating, "We will never understand the phenomena of development and regeneration." And in a survey of the Planarian surgical literature, the investigator Bronsted concluded that Planarian regeneration displays such an array of astounding phenomena that they could very well force one to believe it is accomplished by witchcraft. Now, we have at our disposal, for the past 10, 15 years or so, a set of molecular tools that allow us to ask the question for the first time, how does the Planarian genome control regeneration? And today, I'm going to tell you about some of our work on regeneration and focus on two questions that are important to address to understand how animals regenerate. The first is, if they can regenerate all different cells of their body, where are these new cells coming from? What cells make the new material? Second, if you have a source for new material, what instructions then tell the cells what to make? How do the animals know what part of their body is missing? Now, though we're beginning to get insight into how these processes work, we're still in a beginning phase of understanding the molecular and cellular underpinnings of regeneration. But I'll tell you about what we've learned so far. So first, the source of new material. So in all animals that have many different cell types, like planarians and ourselves, the cells that are different from one another have the same genome, such as a neuron and a muscle cell. What makes these cells different from one another is the set of genes that are on or off in these different cell types. Imagine, for example, these hypothetical two different cell types. These cells might be different by having a set of different genes on or off. For example, in this example, gene 1 is on in cell type A, but off in cell type B. Whereas gene 2 is off in cell type A and on in cell type B. Some other genes might be on in both cell types. Now, what do these genes do in these cells? So each gene is going to produce, it encodes a corresponding mRNA molecule. So gene 1 will encode an mRNA molecule, call it mRNA1. And this encodes, in turn, a protein. I'll call it protein 1. Okay. So gene 1 encodes protein 1, and then proteins carry out the action encoded by the genes. They have all kinds of functions in cells. We can label the mRNA for a gene or the protein encoded by a gene. For example, we can use a fluorescent probe or an antibody that binds the protein, or a fluorescent probe that binds the mRNA. And we can do this with specific probes that label only the mRNA for gene 1, or only the protein encoded by gene 1. For example, imagine we had an RNA probe in red that recognized the mRNA for gene 1. If we added this to the animal with suitable molecular methods, what would happen? Cell type A would turn red, but cell type B would not. Now imagine we added as well an RNA probe to the mRNA for gene 2, and this was green. What would happen? Cell type B would turn green, but not cell type A. And in this way, it is possible to identify the location of expression of any desired gene within the animal. We can also use these types of probes as tools to mark cells and study their regeneration. Let me give you several real examples. So first, here, this is an entire animal with the head at the top, and labeled are the cells of the Planarian kidney-like system called its protonephridia. And there are 3 different probes combined, in yellow, green, and purple, with yellow cells here, tubules in green, and accessory cells for this kidney-like system in purple. Here is an antibody that recognizes a protein that is expressed in the planarian muscle. This is an entire planarian. Here's its mouth. And the muscle is all around the animal. Here are 3 probes that are recognizing cells of the planarian eyes. So here are the 2 eyes of planarians. Okay, so on the left in this eye, in green and red, are the light-sensitive neurons of the eye. And in blue are the pigmented optic cup cells that give the animal this crossed-eyed appearance. Now, labeled here is the nervous system of planarians. This is the brain. Here are 2 nerve cords. Each fluorescent dot here is a single cell. Final example, here's the planarian intestine. Okay. So in this way, we can identify location of expression of genes and look inside the animal at any internal anatomical feature or cell type we desire. Now turning to the cells that are involved in producing new tissues. First, going back to a statement I made at the beginning, a small piece of a planarian, such as from the side, can regenerate an entire planarian. It would include brain and eyes and pharynx and all of these other types of cells I just showed you. So these small body fragments show very broad tissue production potential. Obviously, there's no eye cells within this piece. So where are the eye cells coming from within this piece, and so on. Now, we can consider multiple possible sources for new cells. And I'll go through a couple of hypothetical processes that could occur in this slide. First, let me define a term, differentiation. That is the process of a cell becoming specialized into a specialized cell type. So a specialized cell type then would be called a differentiated cell. So a neuron or a muscle cell, those would be examples of differentiated cells. OK, so in the first example, hypothetical situation in which new cells could be produced, we have a stem cell. So the minimal definition of a stem cell is a cell that divides and that is capable of making more stem cells. Okay, that's cartooned as this circular arrow. And that cell is also capable of making one or more cell types that are different from the stem cell, such as a differentiated cell. Okay, so that's a stem cell. So there could be stem cells that make new cells in regeneration. There could be dedifferentiation, in which a specialized cell, in response to injury, changes, becomes a cell that can start dividing, making more cells, and then redifferentiate to replenish the animal with more of the missing cell types. Or there could be transdifferentiation, in which one cell type that's specialized changes into a different differentiated cell type, the missing one. So what happens in planarian regeneration? Well, it's been known that there is a dividing population of cells in planarians for over a century. And these cells have eventually come to be called neoblasts. So I refer to neoblasts here as the dividing cells of adult planarians. This is an entire planarian, and the neoblasts are labeled in red. They're scattered throughout the body, except for in 2 locations, the tip of the head and the pharynx. And blue in this image is simply the DNA of every cell. So each dot is a cell and the DNA within it. You can see the brain and pharynx quite easily this way. OK, so what this means is that the red cells are capable of dividing to make more cells, but all of the other cells do not do that. The skin does not do that. The brain cells do not do that. The neoblasts are the dividing cells. And the neoblasts are required for regeneration. So shown here are the neoblasts in purple. If you irradiate the animal, the neoblasts, with a suitable dose of irradiation, will die. Okay, the dividing cells are uniquely sensitive to irradiation. And the neoblasts are no exception. They die in irradiated animals. And irradiated animals are incapable of regenerating. Seen here is an animal lacking its head. It never makes a blastema in the absence of neoblasts. Now, the neoblasts are not simply a reserve population waiting for injuries to become deployed, but they're always dividing. And what they're doing is producing progeny cells that go out and replace aging differentiated cells. So as a neuron gets old or a skin cell gets old and dies, it's replaced by a young cell from the neoblast. In fact, the planarians are in this constant state of replacing their old cells of their body. And you can tip the balance between new cell production and cell loss by simply starving or feeding the animal. So if you starve an adult planarian, it starts shrinking. And this process is often referred to as degrowth because it involves a loss in cell number. So the brain is losing cells, but all the while maintaining its form and function. And then you add food, and it can grow back. This requires the neoblasts. Labeled in red in this image are neoblast progeny that are actively replacing aged cells in the tip of an animal head. If you eliminate the neoblasts, not only do the animals fail to regenerate, but they also fail at this process. They ultimately die because of a failed process of tissue turnover, curling, losing their heads, and then lysing. So they require the neoblasts for the maintenance of their adult tissues. And this is a process that happens in essentially all long-lived animals. It's in fact estimated that about 10 billion cells will be produced in an average person per day.
10 billion?
Billion. It's about your body weight in new cells per year. Now, so there is a dividing population of cells that's required for regeneration. And we considered the following two possible explanations for how this might work. In the first possibility, there are cells within the population of dividing cells, neoblasts, that have the potential to make many different types of adult differentiated cells. This type of attribute for a cell, the potential to make many different cell types, is called pluripotency. An alternative model for explaining regeneration would be that the neoblasts have many different cell types that have limited potency. For example, as would be the case if there was a neoblast that could only make neurons and a neoblast that can only make muscle, and so on, and that they acted in aggregate to allow the animal to replace everything. So to distinguish between these possibilities, we reasoned it was critical to look at the potential of single cells from the neoblast population. And the way we initially did this was to use irradiation. Okay, this is a cartoon of the normal neoblast distribution as shown on the left. And as I showed you, if you hit the animals with a high dose of irradiation, the neoblasts will all die. But if you hit them with the right type of dose that kills almost all of the neoblasts, but not all, a few sparse neoblasts remain behind. And this works. And what happens is these individual sparse neoblasts can then start dividing and locally make more neoblasts. Here's an example of an animal that has 4 total neoblasts all together in a cluster. And this is an animal that was analyzed 7 days after the irradiation. And a variety of experiments demonstrate that this cluster of cells arises from the division of one cell. Okay, so one cell divides to make 2 neoblasts, 4 neoblasts, and so on. And these clusters can continue to grow in these local patches of cells. Here's an example of a slightly bigger patch at a later time point, a cluster of neoblasts derived from one neoblast. And these are in red, near the brain of a planarian. And so we can analyze what the cells in these clusters make, and we find that they can make many different cell types, such as neurons and intestine. So what we conclude is happening is that one cell divides to make many neoblasts, and that different cells within this growing cluster of neoblasts make many different cell types, such as neurons, intestine, and other cells. Okay, so this population had the potential to make many different cells, and therefore, by inference, the founding cell had the potential to make many different cell types. We can take single cells that have these attributes. We call these clonogenic neoblasts, or C-neoblasts, the capacity to make clones of cells like itself. And we can transplant single clonogenic neoblasts into the tail of a lethally irradiated animal that lacks all cell division. lacks all neoblasts. Here's an example of a single transplanted SMED1 positive cell in the tail region of a lethally irradiated animal. Has no other cell division. And under optimized culture conditions, with animals that can live 40 to 50 days without cell division, some of these transplant recipients can survive and regenerate. So here are some examples. So animals after irradiation lose their heads, tissue regresses back, they start looking sick, but some of them, after receiving a transplant, come back. Regenerate a new head with eyes, intestine, pharynx, and can eat. And we conclude from these observations that there exist pluripotent adult stem cells in planarians that we call the C neoblasts that explains their ability to produce all of the missing tissues. So how do these neoblasts know what to do at wound sites? How do they know, for example, if they're missing a head or their tail or the tip of the head or their side, as someone asked? How do they figure it out? Well, to explain how we approach this problem, I'll begin with a metaphor. So imagine you want to understand how some complex machine works like this engine. The approach we take is an approach of genetic analysis, where we seek to identify and perturb the component parts of a system. So one approach to understanding how this engine works is to identify and characterize all of the component parts of the engine, and then go in, take one of the parts and break it, and ask, without that one part, what goes wrong in the functioning of the system. The goal then is to infer something about what the function of that one part was for the system. And so for us, the component parts are the genes for this part of the talk. And what we want to do is identify all of the genes and then go in and break individual genes and ask what goes wrong with the system, in this case, regeneration. So the Planarian genome has been completely sequenced, giving us a complete parts list of the genes. We can thus identify and characterize the parts, the genes. And then we want to infer something about the function of that gene. And we do that by breaking the genes using a method called RNA interference. RNA interference is a very powerful experimental tool. And the way it works is it utilizes a molecule called dsRNA, or double-stranded RNA. And double-stranded RNA can destroy the mRNA encoded by a gene, thus shutting down the function of that gene. It never makes its protein to carry out its function. Now, importantly, this method is specific, meaning we can add double-stranded RNA for gene 1 and shut down gene 1 and only gene 1, or we can add double-stranded RNA for gene 2 and shut down gene 2 and only gene 2, and so on. Now, one way we do this is to deliver the double-stranded RNA by feeding. And this was a method first devised for use in a different type of worm, a roundworm, a nematode. But this works in planarians. And what we do is we use bacteria, like E. coli, as a factory to make planarian double-stranded RNA, corresponding to one planarian gene. And then we mix these bacteria with blended liver, which planarians love to eat. And so then they take in these little pills of bacteria along with the liver, and this delivers the double-stranded RNA throughout the body of planarians. And it shuts down that corresponding gene in every cell of the planarian that ate this food. Okay, so in this way, we can go and systematically break genes one at a time, cut the animals, and look at what happens. So when I was a postdoc working with Alejandro Sanchez Alvarado, we performed the first planarian RNAi screen, where we systematically inhibited genes, and we inhibited over 1,000 of them. And we identified hundreds of defects. And I show here just a sampling of the various types of defects. For example, inhibition of some genes blocked regeneration. Inhibition of other genes caused abnormal blastemas, such as pointed blastemas, or blastemas with pigmented freckles on them. Following inhibition of some other genes, we saw growths of tissue or lesions on the body, stretching of animals, blistering, and so on. So we can reveal a huge array of defects that tell us something about what that gene does. Okay? Importantly, the majority of genes that are associated with defects following inhibition have human counterparts. So we can learn a lot about what those human genes do by studying them in planarians. This touches upon an important concept in experimental biology. In the lab, it is frequent and common to use model organisms to study important biological processes of a huge array of types. And studies in model organisms can reveal important and evolutionarily conserved processes. For example, here is a very simplified animal phylogeny, which we have humans and some genetic model system studied in the lab. So if some process or gene of interest appeared back here in the course of evolution, in the common ancestor of humans and this organism studied in the lab, then the process may very well still be present in humans in this model. And we can learn a lot about it by studying it in the model. So for example, most genes we look at that are important for planarian regeneration, they're also found in humans. Those genes appeared way back here. So what are some sample functions for genes that would be important to identify to understand the molecular basis of planarian regeneration? Well, you might imagine some genes are important for stem cell maintenance, stem cell differentiation. We could identify those by studying the planarian neoblasts. And this works. So we and other labs have identified a number of genes involved in regulating planarian neoblasts. And I just showed a list of genes. The identities and names of these are not important for the purposes of this talk. But the point is, we can identify a lot of genes that are involved in the ability of one neoblast to make many other neoblasts, or the ability of a neoblast to differentiate. So these genes were studied in the C neoblast, but some of them were previously known from prior studies. Okay, some genes might mediate wound responses or the initiation of the regenerative process. And we can identify a lot of genes conserved with humans and other animals that are activated by wounds. So here are two examples. This animal's been amputated at these two locations. And then we looked at this middle piece. Seen in blue is the expression location for these genes. This gene's expressed at both wound sites, so is this one. Turns out this second gene is expressed in the neoblasts in response to injuries. Now, some other genes might act to turn stem cell progeny into specific differentiated cell types. And we can identify genes involved in these types of processes. Here's one example. So these are regenerating eyes in red. These other cells descending back from the eye are progenitors for the eye. And they're being labeled by the expression of this gene, SP69. And this gene is required for the process of regeneration of optic cups. Finally, some genes must act in the process of determination of what's missing. And that's what I'll focus on for the remainder of the talk. So what genes specify what to regenerate? And I'll focus this topic on a particular question. And that is, how do animals decide at wound sites whether to regenerate a head or a tail at a transverse amputation? So here's a thought experiment that illustrates the problem that we're trying to understand. So imagine two hypothetical amputation planes, 1 and 2. You can produce a fragment following amputation at plane 1 in which the cells circled in green become involved in head regeneration. Or you can produce a fragment by amputation at plane 2 in which the cells circled in green become involved in tail regeneration. Okay, so the same cells can make either a head or a tail. What matters is not the identity of cells at the wound site, but the identity of what's missing. To get insight into how this works, we inhibited genes with RNAi and looked, following amputation, at these middle pieces that have to regenerate a head on one end and a tail at one end. And we looked for defects in this process. And we found a defect. So following inhibition of this gene, beta-catenin, we see that animals regenerate two heads instead of a head and a tail. So a head on one end, and then in place of a tail, another head fighting to pull the same body in the opposite direction. So a pretty remarkable phenotype, or defect. And we've wondered whether this gene, beta-catenin, is acting just to promote tail regeneration, or whether this gene, beta-catenin, was acting at all wounds to prevent head regeneration. So to distinguish between those possible interpretations, we did the following experiment. So we inhibited the gene, beta-catenin, and then removed the tail and made other amputations in the sides of these RNAi animals. And that resulted in this six-headed animal you see here, which when I saw it was the most dramatic animal defect, was commonly called a phenotype, that I had ever laid eyes on. Question?
Going back to the double-stranded RNA, are there combinatoric effects when you disable multiple genes that produce defects that didn't occur in each of the individual cases?
Yes, and I could give you a specific example in a couple of slides where we have done that type of experiment. But it is possible to inhibit more than one gene at a time with RNA interference. So we can inhibit probably up to 3 or 4 genes at a time. And in some cases, you just see combinations of defects. And in other cases, you might see a genetic interaction. Okay, so beta-catenin acts to prevent head regeneration. How does it work mechanistically? Well, beta-catenin is part of a process called cell-cell signaling. So lots of cells communicate with one another in the body. And the way they do this is to make proteins that secrete from one cell and then bind another cell. And that binding has information. So the signaling process that beta-catenin is part of is called Wnt signaling. In this process, a protein called Wnt is produced from one cell. It's secreted. And this can bind to a receptor on another cell. If this happens, this can activate beta-catenin protein. And what beta-catenin does is it regulates gene expression. Okay, I talked about in the early parts of this talk how some genes are on in some cells and off in others. How is that regulated? Well, there are proteins that throw those switches, turn some genes on and turn other genes off. Beta-catenin helps mediate that type of process. It regulates which genes are on and off. Okay, so to understand how beta-catenin works in regeneration, we must then understand how Wnt proteins are acting. And there are multiple Planarian genes that encode Wnt proteins. And it turns out these genes are turned on, they are expressed in the tails of animals. Multiple of them are expressed in the tail. I show 3 Wnt genes that are expressed in the tails of Planarians here. This one expressed in the tip of the tail, in dark brown here. This gene expressed a little bit further up in the tail, and this one expressed in about half of the animal, emanating from the tail. And I'll tell you only about one of these genes, Wnt1. So Wnt1 is key for understanding how this process works. Again, it is normally expressed in just a few cells, each of these dots, at the tip of the tail. We looked at what happens to Wnt1 activity during regeneration. And first, looking, for example, at an amputated headpiece, we see at the wound that is produced, which is going to regenerate a tail, Wnt1 gets activated. So you see a lot of these purple spots. Those are cells that turned on in response to injury Wnt1. A tail-facing wound is called a posterior-facing wound. So we see Wnt1 is activated, and then with time, expression coalesces down to a new spot at the tail, tip of the tail. All right, so the idea is Wnt's are promoting tail regeneration, inhibiting head regeneration. So this is a nice result. Wnt's are activated at wounds in which a new tail is going to be regenerated. However, to our surprise, we saw that Wnt1 is also activated at wounds in which a head is going to be regenerated. Okay, so this is an anterior-facing wound, a wound that's going to regenerate a head. And Wnt1 is also activated to be expressed here. But its expression does not stay on with time, unlike the case for the tail. In fact, we see that every type of injury we inflict upon the animal induces the expression of this gene, Wnt1. We conclude that Wnt1 is generically activated by wounds. And this raises a puzzle. All right, so first thinking about the posterior-facing wound. The idea is that wounds turn on Wnt1, and Wnt1 promotes tail regeneration. But what happens at the anterior-facing wound? Wnt1 is turned on, but instead of a tail regenerating, a head is regenerated. What makes the difference? So there's one more gene that is critical for understanding how this switch is thrown. And that gene is a fascinating gene called NOTAM. It's a gene found throughout the animal kingdom. Humans have this gene. Humans have Wnt signaling. And this gene, NOTAM, is expressed oppositely to Wnt1. Wnt1 is expressed at the tip of the tail. NOTAM is expressed at the tip of the head. Now, if you amputate the animal, what we see is that NOTAM also gets turned on at wounds, but preferentially at wounds that face where a head will regenerate, anterior-facing wounds. So you see much stronger expression of NOTAM at this site than this site. Biochemically, NOTAM is a candidate inhibitor of Wnt signaling. OK, so we can now inhibit NOTAM and ask what happens in regeneration. We can inhibit it using RNAi. So remember, if we inhibit Wnt signaling, we get two-headed animals. What would happen if we inhibit NOTAM? NOTAM is a candidate inhibitor of Wnt signaling. We would expect there to be more Wnt signaling. What type of defect might we expect to see? Any predictions? Someone speak a little louder. Two tails. That's exactly what we see. Two tails. So this is a NOTAM RNAi animal with, instead of two heads now, two tails. And a variety of experiments indicate, indeed, that NOTAM inhibits Wnt signaling. Turning back to the question asked earlier, if we inhibit both NOTAM and beta-catenin with RNAi, we could ask who wins. It turns out you get two heads in that case. That type of experiment can allow one to infer something about genetic relationships between genes that interact with one another, regulate one another. So this data suggests that NOTAM inhibits Wnt signaling at anterior-facing wounds. Let me summarize these data into a working model for this process. So if Wnt signaling is on, you get a tail. If Wnt signaling is off, you get a head. At amputation planes, wounds activate Wnt signaling. Wnt1 is turned on by wounding. Then, at wounds that face the old head, NOTAM is activated. NOTAM then inhibits Wnt signaling, shutting the process off, and that allows head regeneration. So this is our first known molecular switch that controls a decision made at wounds. And we hope that study of planarians will allow us to learn a lot about decision-making at wounds, about how the decision to regenerate appropriate cells is made. Let me conclude with a summary of some of the key points from the talk. So we believe that planarians are a new and powerful molecular genetic system for studies of lots of important processes in biology, including stem cell biology, tissue turnover, and regeneration. And it is a powerful system because of a number of important attributes, including the ease of culture in the lab, a complete genome sequence, the capacity to inhibit any gene desired with RNA interference, and the presence of many markers for cell types and assays for studying gene roles, such as those examples I've shown you. I also focused on a couple of results that give us some insight into how regeneration works mechanistically. First, we conclude that there exist clonogenic neoblasts that are pluripotent adult stem cells in planarians that underlie the ability of these animals to regenerate. Second, there are molecular decision-making processes activated by wounds that control regeneration. In particular, I showed you that Wnt signaling is a process that is activated at wounds and that it is regulated by a protein called NOTAM to control the head versus tail regeneration decision made at wounds. With that, I'd like to acknowledge a number of people. First, I'd like to acknowledge all of the current members of my lab for all of their contributions to our work, as well as a couple of prior members from the lab who have departed. I'd like to highlight some people whose work I focused upon. First, Dan Wagner and Irving Wang were involved in the clonogenic neoblast work. Dan Wagner, together with Jacqueline Ho, a former technician in the laboratory, performed the work identifying genes that control neoblast attributes. Danielle Venemoser identified the genes induced at wounds. Sylvain Lepan identified the genes involved in eye specification and regeneration. Finally, Chris Peterson was involved in the work of regeneration polarity, identifying the important roles of wound-activated Wnt signaling in the process. With that, I'd be happy to take any further questions you have.
What regenerating capacities do human beings have?
Well, regeneration as a term can encapsulate a lot of different processes. As I mentioned in the talk, each human is going to produce billions of cells in a day, and some of that can be considered regenerative, replacement of damaged or missing cells or aged cells. Furthermore, if you think about all of the injuries you have had over your life, from cuts on your hands to twisted ankles, it's pretty amazing what the human body is capable of. If you compare ourselves, for example, to a car, we last a lot longer. So there's a lot of repair that is going on. It's pretty remarkable what the human body is capable of. There are some tissues that are highly regenerative in humans, like the liver. You can remove part of the liver, and the missing part can grow back. There's some regenerative repair of muscle. There's regenerative repair of skin, intestine lining. So all of these types of regenerative repair processes happen in humans, but the process of wholesale organ production or limb production does not happen. There is a lot of tissue replacement and repair that happens, but it's limited compared to some of the feats that other animals in the animal kingdom are capable of.
Thank you.
You're welcome.
How long do the planarians usually live in normal, and how long do they live once you've amputated them into having 6 heads or 2 tails?
Right. So planarian lifespan is a tough thing to pin down. So the animals are very long-lived. First, if you think about the asexual animals I talked about, these animals are propagating their adult tissue essentially as long as this asexual strain will exist on the planet without ever undergoing sexual reproduction, without ever going through sperm and eggs and producing an embryo. So the adult tissues are being propagated for incredibly long periods of time in these animals.
Incredibly long means?
No one knows. Could be immortal. Somehow I doubt it, but it could be that the asexual exclusive strain is slowly accumulating mutations that eventually are going to lead to that strain's demise. But it could be that strain is going to live for thousands and thousands of years. If you look at sexual animals, these animals don't pull themselves apart and rejuvenate by regeneration. And those animals can live for many years in the lab. I've never seen one clearly age with a process that would convince me it was aging. They do get sick occasionally from infection, but I'm not sure I've seen one age. Now, the recipe for long life would be to have some type of cell that's capable of long-term propagation of the genome from cell to cell, just like the cells that produce sperm and eggs and us do. It's called the germline. Planarians have a cell like that, the neoblast. It can propagate the genome safely for countless generations of cell division. And that same cell is involved in the replacement of any old cell in the body. So they produce progeny cells, neurons and skin and so on. Those cells age, and then when they get old, the neoblast makes a new young one to replace it. So this is a nice recipe for long life that planarians have. Tissue turnover with new cells replacing old ones is essential to any long life, and planarians are masters at this process. Just as an example of the remarkable potential of these cells, I mentioned we have tens of thousands, if not hundreds of thousands, of asexual animals in my lab. All of these animals, as well as the animals of other labs across the world, are derived from one animal. by just serial amputation and asexual reproduction.
So I'm clear on it. You don't see aging in your lab animals.
I have never seen aging in these animals. Now, I would imagine if you were able to take the sexual animals out for enough years under careful enough observation, you might see aging, but I have not seen it.
I wanted to ask, do you guys have any idea like what might be upstream of NOTAM? Like the signal that you saw was promoting head growth, and also especially in the sexually producing animal. Like, do you know like how head and tail end up being specified in the first place?
It's a good question, and so we know that NOTAM gets activated at anterior-facing wounds, but not posterior-facing wounds, but we do not know why. What makes those two wound sites different from one another with regard to NOTAM activity? We don't know. But with regard to what's upstream of NOTAM, we do know one thing, and it's somewhat counterintuitive at first glance. It turns out the upstream of NOTAM is Wnt signaling itself. So Wnt signaling is required for activation of NOTAM. And then NOTAM inhibits Wnt signaling. This type of process is called feedback inhibition. So we think Wnt's at anterior-facing wounds turn on their own inhibitor to shut the process down.
We'll take a question up at the front mic.
So, oh, did you mean me or
Go ahead.
someone else up
Yeah,
the
go
front? Okay. So you showed some pretty cool things with Wnt and NOTAM in relation to posterior and anterior facing wounds. What about when you take 10,000 cells just from some random part of the animal? How does that ball of cells know which end is or was posterior or anterior?
Well, I would posit, though we haven't studied it in great detail in a piece of that size, that Wnt signaling and NOTAM activity would be involved in that piece as well. So at every type of injury we look at, we see this regulation of NOTAM happening. So at that little small piece, at the anterior-facing wound, NOTAM would get activated. At the posterior-facing wound, Wnt would get activated. And that it would know its new orientation based upon its old orientation, and it would control the process through a Wnt NOTAM signaling-based mechanism. But I'll mention also that these small pieces have a lot of problems to solve beyond just figuring out where your head and your tail is. They have to make a lot of the rest of the body. They have to figure out they're missing their side. They have to make more back and belly and many different organs. And there are other signaling processes happening in these types of pieces in addition to Wnt signaling to mediate these types of processes.
Hi, I was just wondering, you said that the NOTAM gene is common in the Planarians and the humans, and I was just wondering when the NOTAM gene is activated in a human, like it's obviously probably related to regeneration properties, but I was curious as to like what kind of regeneration within humans.
It's a fascinating possibility. I would hypothesize that it is going to be involved in many contexts in humans in which Wnt signaling is active. We think it's a feedback inhibitor of Wnt signaling. A reasonable hypothesis thus would be wherever you see Wnt signaling, or in many cases in which you see Wnt signaling in humans, you'd see NOTAM at play, shutting it down or tuning it down. No one has looked, so no one knows where NOTAM is active in humans. But Wnt signaling is active at a variety of type of injuries, can be important for tissue repair, skin biology, bone healing, wound repair. So there's lots of contexts in humans in which Wnt signaling is active. And I think it's a good bet that NOTAM would be present and regulating some of those processes.
Yeah, hi. One of the natural questions that you might want to ask is how, when you split these, have you guys looked at the alignment or the sequence and whether they're exactly the same, or whether you start to see any vast changes in the sequence or alignment?
Well, when we have done a lot of sequencing experiments of a variety of types over the years. And what I can say is we don't see any dramatic evidence for change in gene composition or sequence of genes with time. That said, it's always possible that neoblasts accumulate mutations, and there's some subtle heterogeneity within the neoblast population. But we have never sought to examine that. Yeah.
I'm curious about whether these organisms contain cells that interact other than through sending their signals externally. That is, through gap junctions or some other way? And if so, have you looked at that to see if there's any sort of feedback or any organizational systematics that go on there?
We have not studied gap junction proteins in my lab, but there are gap junction proteins in planarians, and a number of them have important roles in regeneration. So that is, those are present, and they are involved in regeneration. But the details of how those genes are mediating regeneration is unknown. But it's possible that there's some communication, not through secretion and binding a receptor, but through cell-to-cell communication through gap junctions as well.
So we'll take one last question.
I'm curious, how does, when you have a 10,000 cell patch and it's missing all its systems that it normally would have, how does it have the energy and all the things required for it to grow?
Great question. I'm glad you asked. You know, this touches upon a very commonly misunderstood attribute of regeneration. It would seem to defy the law of physics that a 10,000-piece animal without a mouth and without a brain could regrow all of the missing cell types at the same proportion. Where would it get the energy? Where would it get the mass? In fact, it doesn't. It can't regenerate all of the missing parts of the body at the same scale. Instead, what it does is it regenerates some structures at the wound sites. Let's take a tail fragment as an example instead of a 10,000 cell fragment. A tail fragment regenerates a head at the anterior facing wound. And then in the middle of the old tail, a pharynx appears. The old tail tissue is becoming transformed to become mid-body-like. In fact, the internal anatomy is getting rearranged in this process, a process called morphollaxis. The end result is a small animal that has transformed to have a complete complement of organ systems at the right proportions, but all smaller than was present in the original animal. That animal now has a brain and a mouth, can eat, and grow to be the size of the original.
So before I invite you to the reception, let's give Peter one more big round of applause.